Cool. So we need,
Yes.
and are you comfortable with how we took not the male, well, we'll do that to him too, how we took the female and put her on her side? Usually you're comfortable with the female anatomy because you've seen my, wonderful drawings. But I mean, it's basically the same structure. Here we go. Usually this one makes a lot of sense to you because I've drawn this rudimentarily, but I've still drawn it for you literally for years. The only addition here is I need to be sure that you're comfortable with the ovarian ligament. That this is not a duct. This is strictly connective tissue to anchor the ovary and make sure that the ovary does not float around the female's abdominal cavity. Because where the oviduct opens and where the ovary is, that is proximity. They are not physically attached. And so the ovary has to be held in place by this connective tissue, almost like a kite on a string, because teenagers fly kites a lot now, right? All right, so then we took her and we cut her in half. So this is her abdomen here. This is her buttocks here. So she's facing to the right. So her belly button would be right about there. Okay, and so you have these two structures. You have the urinary bladder and the uterus right on top of each other. But this is also why when males get to the last 3 months of their pregnancy or the last trimester, why she has to urinate all the time, okay? Because about month 6 or 7, the baby's had a lot of space to move around. And about month 7, the offspring goes head down, and the crown of the head sits, is that other diagram? The crown of the forehead sits right on top of the cervix. I mean, right here, sits right there, okay? And then the baby starts to grow a lot. And as the child increases in size, the uterus starts to push more and more and more on the bladder until in month 9, she hits in her gestation about week 33, 34, 35. The bladder gets reduced down to about the volume of a golf ball, like about that big, okay? And so women in their last trimester, they have to urinate all the time because they are consuming large amounts of fluid, and then they're processing that fluid, and then they've got to pass it. Okay, so it's an interesting anatomical orientation in that the uterus sits on top of the bladder, but obviously it didn't impact their evolutionary fitness. So while it's really uncomfortable, from what I understand, what I've been told, it didn't impact our evolutionary fitness. So the structure stayed. All right, so from then the female structures, then we simply slide into 6F, which will be the male structures. And usually those are less familiar because we don't link the male anatomy to any content before this. So this would be 6. This would be 6F. Let me double check my time. All right, so this is the male reproductive system. Okay. And there's going to be 7 structures here. There were, for the female, I think there's 6. Yep. So for the female, there's 6, and for the male, there are 7. So 1, 2, 3, 4, 5, 6, and 7. So the testes or the testicles. Then there's the epididymis. I should slide that up. Sorry about that. Let's get rid of this too. E-P-I-D-I-D-Y, yep, M-I-S. That's pronounced epididymis. Then the vas deferens. Oh, I made it through all of first period. I didn't make a mistake. And I thought, let's see if I can do it during first period or second period. Nope. Okay, the vas deferens. You're actually familiar with the vas deferens. You just don't know it yet, but you will. R-E-N-S. Now, technically, that's vas deferens, but most people, when they pronounce it, they drop the first E and they just pronounce it vas deferens, but it's really vas deferens, kind of like Wednesday. Then the urethra. And then we have the seminal vesicle. and we have the prostate gland. And they have the largest name and the smallest structure, the bulbourethral gland. So this big old name for a structure that's smaller than the last digit on your pinky. It's this little gland, big old name. Where's my red pen? And then I want to slide in on the side. These three I put over here intentionally, because these three produce about 95 to 98% of the semen volume. The material that the male ejaculates during sexual reproduction, that whole fluid is called semen. And so people talk about, oh, the semen, it's usually 5, 6, 8 mils, depends on the male. And people talk about that containing sperm. And what's fascinating is less than 5% of that entire fluid is actually sperm. Okay, it's water, sugars, and buffer. In fact, semen is 95 to 98% water, sugar, and buffers. Okay, it's actually a very, very small volume that's actually the sperm. But that small volume is impressive. Okay, that only 2 to 5% of the volume holds on average 600 million sperm in one semen sample. Okay, that's why the pull-out method doesn't work, just saying. Okay, so to wrap your mind around that, in the United States, there's about 350 million people, about. Okay, so if you take the number of people in the United States and double it, okay, I'm not talking San Diego, I'm not talking California, in the United States, take the number of people in the United States and double it, and that's how many sperm are in one semen sample. It's impressive. Okay. So let's take that male from the front. Where's that male diagram? The diagram's over here. Over here, may I take this? Does anyone still need this? It's a better question. All right, and let's move that out of the way and slide this all the way. Oh, come on. Like that. Good grief. There we go. There's the male. Looking at the male from the front. Now, what makes male sexual reproductive system interesting is that part of his reproductive system is internal and part of his reproductive system is external. Okay, that is quite different from the female, where the entire female reproductive system is internal. Okay, parts of the males are external for the transport of the gametes, the transport of the semen. And also the testicles are external for temperature control. And we'll talk about that in just 1 minute. So let's go left to right. So I don't know how well this comes across on your camera, but you should see the shadow of some tissue here. And that is the scrotum. And so the scrotum is an extension of the skin. And there's connective tissue that can move the testes up and down, either close to the male's body cavity or far away, and that's for temperature control. Sperm are active or viable for about 7 to 10 days. We don't call sperm alive. They're either, you can call them motile or mobile. So you will hear about the motility of sperm. You will hear about the viability of sperm, the activity of sperm, but we do not call them alive or dead because they are not separate organisms. They are cells that are either mobile or not mobile. So temperature, sperm are highly temperature sensitive. So the testes have the ability to be close to the body cavity or far away for temperature control. So then the sperm are produced in this gonad called the testy, or the testicle. Good morning. How you feeling?
Better.
Fantastic. Last night, weren't sure if Ariel was healthy or not, but he's better today. That's good news. So this is the testy from the outside. If you take this testy and cut it in half, okay? If you cut the testy in half, it then exposes the structures on the inside, and we'll talk more about those. So then there's a gland draw my lines in red again. Sorry about that. There's a gland that sits on top of the testes. Good grief, the news is even coming in saying, hey, yes, I know that George is counting their votes. Yes, I will check later. There's a gland that sits on top of the testes. This is actually where the sperm, the mature sperm, are stored. Semen is not stored here. Semen is actually produced internally here. The sperm, the cells themselves, the gametes, are stored inside of the epididymis here. Epididymys. That is a fun word to spell. So the sperm are produced inside of the testicle, and then when the sperm are mature, they're called spermatozoa, and then the spermatozoa are actually stored in the epididymis for about 7 to 10 days. If the sperm are then to be used, the epididymis contracts, and that's the beginning of ejaculation. If the sperm are not used, then the male's immune system simply breaks down the sperm and recycles the macromolecules. So let's say that he is sexually aroused, he reaches climax, he ejaculates. The epididymis then contracts, okay? When the epididymis contracts, it drives sperm, not semen, okay? It drives sperm out of the epididymis and up a tube. So the sperm actually start externally and they actually travel inside of the male. The tube that the sperm flow through, but only when he ejaculates, is called a vas deferens. And there are two, one for each side. Fine. The sperm then actually go inside of the body cavity, and there's some tissue that we'll talk about here in just 1 second. So there are glands that are then responsible for the production of the ejaculatory fluid, what we call semen. One of the glands that is responsible for producing part of the semen is the prostate. Gentlemen, we'll talk about a prostate exam probably on Tuesday. I don't think we might have the time today. And we will talk about how the internal organ called the prostate, once you start having physicals, or for the athletes, maybe you've already had this experience, how they have to palpate or check the density of your prostate, give your prostate a little squeeze, or actually it's more of a poke. You'll see. Then you have the urinary bladder here. So this is the urinary bladder. Like such. And so the vas deferens actually, when they go internal, they actually then wrap around the back of the bladder. And then that little gland with the big old name sits right here. Let me change the angle on this. That gland right there, that is the bulbourethral gland. There are two. They are bilaterally symmetrical. Bulbourethral gland also makes contributions to the semen. Once the semen is fully formed, it then enters this tube here, this tube that then carries both urine or semen, depends on what the male is doing, out of the body, and that would be the urethra. Here. So you'll notice how, let me put that back for you. You will notice here how the urinary bladder runs a tube through the prostate, but we just got done saying that the semen flows through here as well. So there's another tube, this diagram doesn't show it, there's another tube that connects to the urethra inside of the prostate, and it's actually the prostate gland that controls which fluid the male is releasing through his urethra, whether it is urine or it is semen. But that should be 1, 2, 3, 4 up the right hand side, 1, 2, 3, 4, and then 1, 2, 3, 4 on the opposite side. Okay, so if this is the male looking at him from the front, what do you think we're going to do to this male? Yep, we're going to take him from the side, we're going to cut him in half. He's not happy about that, but he'll get over it. All righty. Here we have testes. Here we will have, I'll grab the epididymis this way. Grab that way. So there's the epididymis. Then this will be the scrotum. These will be the vas deferens. Yeah, that Sharpie on this camera works really well. So
What about the camera working?
there's kind of an orange, pink, red hue on these handouts when I print them in color. And so I usually, when I do it in black and white, I use the red pen, and the red pen really pops. But since these are in color, the red pen, it kind of blends in. So I'm using the red for the words, so the words will still pop. But then I'm using the Sharpie for the structure. And everything works pretty well. If you guys don't like it, let me know. Okay, here's epididymis. E-I-D-I-mis. For someone who doesn't spell well, that is a struggle to spell that word every single time. All right, then we have the urethra. So you will notice there is this structure. There are two different structures inside of the penis, and these become rigid when they are engorged with blood. Okay, this is in no way an extension of the skeletal system. This is not connective tissue. This whole idea of a bone does not exist, of course. This is tissue that when he is sexually aroused, blood flows to this tissue and it fills up just like a balloon and becomes erect. So then this is the tube, the urethra, like such. And then this handout does a fantastic job showing the path of the sperm. So the sperm, again, is stored, the sperm is produced inside of the testy. When the sperm is then fully formed, so you have what's called the sperm head, and we'll talk about this in core 11. There is then a tail that is attached to that sperm head, and at that point it is considered a mature sperm, and so the name changes to be called a spermatozoa. So once the spermatozoa are formed, or the mature sperm, they are then stored inside of this gland here called the epididymis. Okay? When the male ejaculates, the epididymis contracts. When the epididymis contracts, about 300 million sperm are then released from the epididymis, and they flow through the vas deferens. The only time that the vas deferens have sperm in them is when he ejaculates. Okay, so then the sperm, nice catch, the sperm then flow through the vas deferens, and notice how the vas deferens then goes way inside the body, wraps around the back of the urinary bladder, and joins the prostate. Okay, so the vas deferens is actually a tube. It's about 25 to 35 centimeters long. It depends on the size of the male. Okay, but it's actually quite long. So we got the epididymis, and then it then joins this gland here. This is the seminal vesicle. Oh, that's not acceptable. Seminal vesicle here. Like such. And now we need to be sure. I'm not sure the highlighter will really work here very well. Not really at all. Okay, do you see this Y-shaped structure here? Do you see that intersection? Yes. Okay. So understand that if there's fluid flowing here, that's semen. If there's fluid flowing here, that's urine. Okay. So inside of the prostate, these two tubes, and the name of the tubes now don't matter. There's a tube out of the bladder, and there's a tube after the seminal vesicle, and they both enter the prostate. And where those two tubes join, then that's the beginning of the urethra. Okay. And then inside of the prostate, at the beginning of the urethra, is where the tissue decides whether the semen or the urine is going to flow out. So we will label this structure later. Come on, pen, I need about 10 more minutes. So then this organ that we're talking about here, this gland actually, that is the prostate. Now, as now this fluid would be considered semen. As the semen then flows through the urethra, you have another, I should label this too, label that one later. You have another contribution to the semen here, and that's the bulbourethral gland. Like such. So again, IB will never, ever ask you to draw this from scratch, ever, okay? When they have provided you with the male diagram, it's kind of a 50-50 split, okay? Some years they provide the front view, and some years they provide the side view. However, with the females, I'd say 90-95% of the time, it's the side view, okay? Now, when you study this, be sure that you're studying the blank version, okay? Because that's what you're going to see on the exam. Sometimes when you look, especially with human anatomy and physiology, if you study the completed diagram, you go, oh, I got that. But then when you get the exam, what you see on the exam doesn't match how you study, all right? So be sure and grab 2 copies of this out of our folder. And when you go to study this, study the blank one and be able to name all of the structures. And if you don't remember, then go over, take a look at your completed diagram, but always study blank diagrams when it comes to human anatomy and physiology. All right, so 2 things. The density and texture of tissue tells doctors about how healthy the tissue is. So if you guys ever had a physical and they push on the bottom of your ribs and they'll start asking you questions, and what they're trying to do is they're trying to distract you, right? Depending on your relationship with your doctor, they'll talk, and right, and so then they'll get over here by your liver, right, and then what they're doing is they're feeling your liver, okay? They're feeling the density of it, they're feeling the surface of it, and then they'll ask you to take a deep breath, and they'll ask you to breathe out and relax. And then have you had this experience when they palpate your liver, right? All of a sudden, they go, right? And you're like, oh, what's that, right? Okay. What they're literally doing is they're pushing under your rib cage and they're giving your liver a little squeeze. But doctors don't call it squeezing your liver; they call it palpating. Whatever. They're feeling the density of the tissue. They should feel a uniform density. They should feel a smooth texture, and then different organs. They are trained to know the density of the organs. It tells them about the health of the organ, whether you have any tumors, whether you could have a bacterial or viral infection. And so depending on how the organ literally feels, tells them about the health of the organ. Well, they have to check the density of the prostate, okay? And how they do that is they enter through the male's anus here. And because this large tube right here, just like the female, this is the rectum. It's the end of the large intestine, okay? So what your doctor does then is they will put some lubricant on their finger and they will enter through the anus, and usually they're talking to you here, how about those Padres? How about the Chargers? Do you like football, right? And while they're talking to you, they are palpating, but usually only with a finger just because of size. They are palpating the surface of the prostate. Same thing. They're checking for a smooth prostate, and they're checking for the density of the prostate. As males age, the density of the prostate can change, and that can lead or that can indicate the health of the prostate and whether or not he should be screened for prostate cancer. Okay. Then we talked about surgical sterilization with the female. Yes.
Yes.
Yes. Have we talked about getting her tubes tied? Yes. Okay.
Yes.
Which really, of course, they don't tie the tubes. It's a ligation, right? They cut it out. And so this is the surgical sterilization of a female, all right? You can do the same thing with males, all right? And actually, it is better if any partner is going to be sterilized medically, it's better for the male to be surgically sterilized than the female, okay? Because in order to surgically sterilize a female, you have to go through the abdominal wall, okay? You're getting into the body cavity of the female, and the risk of infection is higher, all right? So the male, right? This tissue here of the vas deferens, do you know how yesterday we were talking about the structure when we are all embryos, the reproductive tissue starts as the same tissue, and then what becomes the uterus in females becomes the prostate in males. But the uterus and the prostate originally were the same tissue when we were all embryos, okay? The tissue for the ovary and for the testes start as the same tissue. The tissue for the penis and the clitoris start as the same tissue, okay? The scrotum starts as the scrotum, and in females, the labia start as the original tissue, Okay, so in females, the oviduct, and in males, the vas deferens start as the same tissue. Okay, so if you need to ligate the vas deferens, what they do is they make about a 1 or 2 centimeter incision in the back of the scrotum, and they then use what looks like a knitting hook, and they go inside of the scrotum and they pull out the vas deferens, and just like the tubal ligation in the female, they ligate the vas deferens, seal each side, put it back inside. All right. They then, of course, do this to both vas deferens. Well, whenever you have something taken out of your body, it's called an ectomy. Okay. If you have your appendix out, that's an appendectomy. You have your tonsils out, tonsillectomy. Well, if you have your vas deferens taken out, that is called a vas. People then run those two terms together, and what do they call it?
Vasectomy.
A vasectomy. Yep, which most people don't recognize, because when we say vas deferens, we put the stress on the A, vas. Okay, whereas when you put vas and ectomy together, they move the stress in the word and they put it on the E, and they call it a vasectomy. Okay, that's where people disconnect. It's a joke. Okay, so the male keeps his testicles. You are not castrating him. Okay, and he still maintains the ability to have an erection, and he still releases semen. Okay, because that's why I wrote in the notes earlier. No, there we go, where we said 95 to 98% of the seminal fluid comes from these internal glands. Yes. Notice how the epididymis isn't listed over here, because only about 2% of semen volume originates in the epididymis. Okay, so if a male has a vasectomy or a vasectomy, he still produces semen. The difference is that the semen that's produced, the seminal vesicle and the bulbourethral gland and the prostate, simply doesn't contain any sperm. Okay, so when he ejaculates, the epididymis still contracts, and the doctors leave enough length in the vas deferens so the vas deferens fills with the sperm, but then the sperm join, or I should say get to where the vas deferens is cut, and they don't go any farther. Okay, and then what he ejaculates is simply a fluid without any sperm. Okay, so that's what a vasectomy is. So that should take care of, wasn't it question 6F?
Mr. Monroe,
Yes.
is the male urinary bladder bigger than the female?
Okay. What my training tells me is no. What real life tells me is yes. So, anatomically, no. But typically, I have seen this, the term for this is anecdotal data, that females tend to be able to hold their urine much longer than female. No, females can hold their urine longer than males, and I don't know why. But anatomically, our bladders should be the same size. Okay. Now, males, as they age, because of testosterone levels, the prostate tends to grow in size as you age anyway, and then it also grows in response to long-term exposure to testosterone. So the prostate then starts to push on the bladder as males age, which is why males over about the age of 40, 45, 50 to 60 have to urinate more often. Basically, men turn into pregnant women. No, I'm kidding. That the males do tend to urinate more often as they age because of the increased size of the prostate. But yeah, females, and we should find a, let's see, someone who studies this would be a urologist. We should find a urologist and say, all right, what's the deal? It sure seems like females can hold their urine much longer than males.
Oh, okay, thank you.
Okay, you're welcome. All right, so that should take care of, that was 6. That was 6E. No, that was 6F. Yes. Okay, so then that would take us up to 6G, and that's a good place to stop, because 6G, we need to talk about the testes determining factor, or the TDF gene, which makes males males. Gentlemen, we are males because of a single gene, the TDF gene.